Today, it is my greatest pleasure to introduce Dr. Alex Martin from the National Institute of Mental Health, or NIMH. Dr. Martin received his PhD from the City University of New York, or CUNY. He is acting chief of the Laboratory of Brain and Cognition and the chief of the Cognitive Neuropsychology section within the intramural research program of NIMH. His laboratory focuses on understanding the neuroorganization of human memory and social processing systems and how they break down in neuropsychiatric disorders. I and probably most of you know of his seminal work in object and concept representation, which he'll be talking about today, but his research fields are very broad and published over 200 peer-reviewed papers that range from autism spectrum disorders to real-time fMRI feedback training and fMRI adaptation. I remember the Trends in Cognitive Science paper with Calanegro Spector in 2006, I believe. That is one of the most cited papers in the field. Dr. Martin is an associate editor of the Neurobiology of Language, editorial board member of Cognitive Neuroscience, and direct submission editor of PNAS. Alex is also an elected fellow of the American Association of the Advancements of Science, the Association for Psychological Science, and the American Psychological Association. And when I was looking at his long CV earlier today, I saw that he already included the BIRC talk in his CV. So thank you for doing this. And this probably tells how exceptionally organized he is as well. I was just looking back at the email correspondence with him, and the first communication of the invitation went out to him back in May of 2019. Then, of course, COVID hit. We had to postpone, and hopefully we were hoping he can come back in person to do a hybrid. But unfortunately, we decided to move ahead and have him speak. So we couldn't wait longer than 2 years, but finally this is happening today. So it is particularly a great pleasure to have Alex today as the speaker. Please join me in welcoming Dr. Alex Martin.
Okay, can everybody see my white screen here?
Yes.
Actually, I'm not seeing you, but let me get out of this. So is everybody's camera off? Is that how you do this?
Some people are off and some people have it on.
Okay, well.
It might be, you might have to change the view options.
Yeah, I'm looking for that. It would be nice to see all those
There's
friendly
a
faces.
little tiny view on the right icon, and if you click on it, you can choose standard or side-by-side gallery.
Yeah. Screen. Huh, I lost that whole screen. But can people see me?
Yes.
Okay, well, good. Let me fire this up. So thanks for the introduction. And the goal really was for me to come visit, and we kept we kept pushing this back, and then finally thought certainly by October of 2021 I'd be able to come, and clearly that did not work out as we planned. All right, before I start, I'm sure, as you all know, we just lost a few days ago Mort Mishkin at the age of 94. In my career, incredibly fortunate to have been able to interact and talk to him for over 40 years. You know, when people talk about how we stand on the shoulders of giants, this is the guy you should think about whenever you hear that. So now we can begin. This was the original title I gave you, which I thought it was a little bit too long and didn't say anything about concepts, which I knew some people would want to really hear my thoughts on. So I simplified it to that. So that's why there were two titles. So let me quickly go over the plan of the talk. It's going to be in 3 parts. The first part, I'm going to talk, let's just give you some general points about how we represent object concepts and their properties. And my goal really isn't to change your mind about anything, but to just give you a sense of the way I think about these issues. Then I'm going to turn to what I really think is one of our obviously most important and also most neglected object categories, and that's food. Why that is kind of an interesting question. And also has been ignored in terms of the senses. It's been taste, although obviously that's changing rapidly for obvious reasons. Then I'm going to turn to the third part of this talk, which is an issue that's going to come out of the studies on food and taste. And this is the problem of overlapping neural systems, which happens all the time in the neuroimaging literature. And I want to talk a little bit about what that means when it occurs and how we should deal with it. Okay, first, a couple of issues. The first issue and point I want to make is semantic memory is not a thing, right? It's lots of different things. It's everything we know about the meaning of objects and words. It's about facts, ideas, beliefs, and so on. It's basically all of your long-term memory that's not autobiographical. And as a result, and I know you've seen probably dozens of papers telling you where the semantic system is. There's no such thing. There really isn't. And when we make that argument, or others have made the argument, this is really a prime example of what's considered a category mistake, which, as I've written here, is when we present something that belongs to a particular category, in this case, a concept that helps us think about memory, like implicit memory, explicit memory, semantic memory, the concept that belongs to a particular category like that, as if it belongs to a different category, like a neural system. What really matters, if you wanted to talk about brain organization for semantic memory, is what domain of knowledge we're talking about. Okay, that's the first point. The second point is that the anterior temporal lobe, the so-called ATL, that a lot of people, and I think some of the people on this Zoom right now, talk about, is not a place. It's lots of places. Here on the left is the human temporal lobe. This is the temporal pole. And the pole itself is divided into 6 cytoarchitecturally distinct regions. Here on the right is one of our recent papers. This was just published in the Journal of Neuroscience, looking at resting state parcellation of the same area. And really our job is to figure out what is the function and what is the connectivity of each of these regions. Okay, so let me continue. I'm going to give you 5 points about concepts in the brain that I'm pretty sure are true. Let's find out. Okay. Point number 1. All concepts are abstract. I got something in that. Okay. All concepts are abstract. And it's not only because they're based on probably the most abstract thing on the planet, which is the neural code, but it's because they're all prototypes. Look, there's no concrete concepts. There are concepts that refer to concrete objects in the world and concepts that refer to abstract ideas, but they're all, as things, abstract. And it says here, that's because they're prototypes, which are summary descriptions of a category as a whole based on properties that are often found in that category. They're probabilistic and they're graded. Here's a prototypical chair that might live inside your head, and that's a chair as well. They're all chairs. And here they're all can openers. So it really is about function. And for these kinds of objects, not form. Point 2, properties are content specific, not modality specific. So here's an example. This is from Hans Optebek's lab. This is a study of sighted controls looking at the ventral stream. Here's occipital cortex back here. So this is up through the fusiform. And this is a game you can play ad infinitum. In this study, they did faces, objects, scenes, and bodies. And you get this very consistent finding of these systems that go across occipital temporal cortex. Here's in the same study, the same representation, the same organization, except these people have never seen any objects. They're congenitally blind. This is one of a series of studies actually going back into the late 1990s. So there's probably a dozen studies like this. So as we've been arguing again since actually 1995, what I think is represented in this region is object form. But what this tells us is that this occurs regardless of the modality that the input came through. If you're congenitally blind and you learn about object form through touch or from hearing, and actually in this case, faces was done not by words, but by listening to people laughing and so on, just sounds. Get the fusiform face area right there. So the modality of input isn't important. What's important is the content of the property. And like I just pointed out, in this case, object form. Point 3, properties are distributed in distinct locations, not stored in a single place in the brain. And they don't converge in a single hub either. There's no semanticizer that lives in this interior temporal lobe. I'm not going to belabor this point. This paper, which came out several years ago, is a page and a half long on, I think, listing all the difficulties with this kind of hub idea for representing all conceptual knowledge. So those who are interested, any of you are interested, you can find this paper in this journal, which, by the way, I had never heard of. And I bet most of you haven't either. Turns out it's the highest impact factor in all of biology. So get a paper in Physics of Life Reviews. Okay, point 4. These distinct locations that we just talking about interact to form a network or a circuit. So here's data from one of our circles, one of our manuscripts, one of our papers. These blue areas are regions that were more active when people were viewing objects like this, a tool versus an object like that. It's the left middle temporal gyrus, interparietal sulcus, and premotor cortex. That's a task-driven representation. And here's the functional connectivity based on, in this particular example, taking this point here. I'm sorry, I hit the board here. This point here, using that as the seed, and here's the actual data from this individual of the connectivity between left premotor cortex, left middle temporal gyrus, and left premotor cortex. We've published this 3 different times. I know lots of people are concerned about how replicable these findings from imaging are. So here's an example from this program called NeuroSynth that I hope most of you are familiar with. It's an automated meta-analysis. You pop in the word tools and it summarized findings from 115 studies, and that's what it generates. So you can be the judge of how carefully or how closely this representation agrees with those. So as far as I'm concerned, all of these, and we can talk about this later, all of these areas are convergent zones. They're all hubs. And finally, point 5, these locations are not randomly distributed. They follow a specific plan based on the organization of perceptual, action, and emotion systems. And what that tells us is that semantics is not smeared across the cortex like cream cheese on a bagel, as certain individuals have argued. One last point, this is a bonus point. Concepts and semantics are not the same thing. You look up the word semantics, I think the only ones who are confused about this are cognitive neuroscientists. If you look up semantics in any dictionary, it'll tell you it's about the study of the meaning of words and sentences. And why this is important is that, well, let me give you an example. This is my dog here. This is Winnie, and this is my latest grandchild, Ruby, who's just 5 months old. Both of them, certainly Ruby and Winnie, have lots of concepts. Neither of them have any semantics. And this is important, at least in my mind, is because if we're going to have an evolutionary story and a developmental story about the nature of concepts, we need to keep that idea separate from semantics and the language system. Okay, I'm going to be using this quote from Freeman Dyson, a famous physicist who actually just died last year, Nobel Prize winner. And this is one of his quotes. There are actually a number of people who have said similar things. New directions in science are launched by new tools much more often than by new concepts. By the way, I just learned that Freeman Dyson never got a PhD and talked and wrote vigorously about why he thought that was a horrible system. So you PhD students can maybe look that up to see what he was talking about. So we need new tools, but we also need ideas and models and theories. You see, I put new in parentheses, because I'm not sure anybody has any new ideas, but we've got to have them to understand the data. So this is my latest and last attempt, or most recent attempt, to try to lay out a model of how we think about object categories and object concepts. This is the GRAPES model, which stands for grounding representations and action, perception, and emotion systems. And the critical point here is that object categories are organized in the brain by their functional, excuse me, and other critical properties as represented in action, perception, and emotion systems. And I've referred to these in the past as conceptual primitives. So look, for this object here, the primitives would be, well, they're round, they're usually green or purple, they're sweet, and they're graspable. As opposed to what we typically refer to as encyclopedic knowledge, which in this case would be, oh, this stuff is used to make wine. They grow in Northern California and Italy, and all your other verbal, explicit knowledge about the category of grapes or any other object. So here's the image I want you to take from that. These are the primitives. And the idea is that they provide this foundation or scaffolding for all this knowledge, this encyclopedic knowledge that we have access to, supported by the conceptual primitives. And the importance of this idea is the argument that this, and through these primitives, this is how mental representations become grounded. And that means this is how they get connected to the things they refer to in the real world. All right, now let's turn to the main category I want to talk about, and that is this category of food. Well, how is that grounded? Well, we know the function. It gives us pleasure, but the main function of food is survival. And the main property that ensures survival with food is taste, because taste allows us to identify whether a food is safe and nutritious or dangerous. So this story started back in 2005. This is Kyle Simmons. And at this time, Kyle actually was a postdoc in Larry Barcelow's lab. And we did this very early study of just having people looking at pictures of appetizing foods versus objects like these buildings. And what we found was there was activity in orbital frontal cortex, shown right here, which we tied to reward and the hedonic value of food. And also in the insula, this region, dorsal mid-insula, that was active. Let me focus in on this thing. So as you can see from the title of this paper, our argument was that pictures of appetizing foods activate gustatory cortices. The truth is, we had no idea if that's gustatory cortex. I mean, there was evidence in the literature, and we showed a picture of the evidence suggesting, taste cortex could be here, but it could be there or any place else in the insula. So we need that new tool, and we built it. This is our gustometer, which allowed us to administer, it's a scary looking thing, but allowed us to administer a tiny bit of a tastant, about 1/6 of a teaspoon, into our subjects while they're in the scanner. Okay, so now Kyle, he's a postdoc in my lab. It's 8 years later, and now we can do that same experiment again, looking at appetizing, at least for some people, foods versus objects, and then map out taste perception to find exactly where in insular cortex was gustatory, primary gustatory cortex. Now, there was another wrinkle to this study. This is Kevin Hall at NIDDK at the NIH, who does studies of food, and he has a ward where we could have precise control. So everybody had a standard meal. I'm sorry why it keeps doing that. Everybody ate a standard meal, and afterwards we drew blood, or they drew blood, so we could measure plasma glucose, and then we did the food perception and taste perception tests in the scanner. And here's what happened. There's that mid-dorsal region of the insula. That's the taste-sensitive cortex I was just talking about. And as shown here, this cortex is responding to pictures of food, as we thought we were seeing back in 2005. But the magnitude of the response was dependent on the amount of glucose circulating in peripheral blood. The lower the level of glucose, the greater the response when viewing objects like these and not like this, these. These are the non-food objects. So that's a significant interaction. So what does that tell us? Well, I think there are two main points. One, that this category of food is grounded in the taste recognition system. And two, the response of this taste-sensitive piece of cortex when viewing food is directly linked to the body's homeostatic state. Well, how does that happen? Well, we have a good idea because here are our cranial nerves. There are 3 of them from our tongue. Here's this system that's been well described, getting information right into this part of the insula. And at the same time, there's massive input into the insula from the cortex, including back here in occipital cortex, the fusiform, up through entorhinal cortex. So dorsal mid-insula is an integrative region. It's not simply primary gustatory cortex. And in this case, it's giving us evidence for the integration of visual information and signals from the body. But why? Why does the insula respond when you just simply view pictures of food? I just showed you evidence that what everybody considers visual cortex responds in a categorical way to viewing objects in the congenitally blind. Yet we always call that visual cortex. Nobody, including me, would call the insula visual cortex, even though it shows this very clear categorical response to viewing foods that's as strong as anything we see in the fusiform. And if that's, if you feel confused, I'm confused too. But that's just the way it is. So why is this happening? Well, I think there are two main possibilities. One, this is a general arousal alerting response, or in addition, it's actually giving the organism a specific amount of information based on the quality of the taste, whether it's sweet, sour, or salty in that example. Of course, to answer that question, we need to know how taste quality itself is represented. So that's the first thing we did. This is Jason Avery. This was published last year, Journal of Neuroscience. And when it comes to taste representation, we got two possibilities, right? You're going to have a spatial map, gustotopy, like retinotopy or tonotopy. And this is in the mouse brain, where there was some evidence reported that looked just like that. Or you're going to have a population code. Again, in the mouse brain, there was some evidence that had been reported where it looked like the neurons are broadly tuned to multiple tastes. So the population will tell the organism what the taste is. So now we're going to turn to high resolution, high magnetic field strength. This is 7 Tesla with these small voxels as indicated here. Everything I'm going to show you for the rest of this talk is done in 7 Tesla. If you just look at univariate analysis, our sweet, salty, and sour tastants, and here's the formula for those of you who are interested, relative to our neutral taste, which is actually known as artificial saliva. There's the formula. And believe me, we never tell the subjects that it's artificial saliva. They think it's water, which is what it tastes like. And when we did the comparison, we got that same region of the mid-dorsal insula for sweet, sour, neutral, or them all combined versus, sweet, sour, and salty versus neutral for that same region. If we now look to see any evidence of gustotopy, that is regions that were selective for sweet, salty, or sour, we really found no evidence. This is the group data. This is just a snowstorm of nothing, nor at the level of the individuals. So here's a subject where activity throughout the insula was biased towards salty, brought that subject in again another time, and everything was biased towards sweet. So no evidence for gustotopy at the group or single subject level. However, using a multivariate approach, multivoxel pattern analysis, we found strong evidence in the sense that you could easily classify the difference between each of the tastants versus the others, salt versus sour, salt versus sweet, and sweet versus sour in both the left and right dorsal mid-insula region. Okay, so now we know, and this is very recent, there's now converging evidence in the mouse. This was just published in Current Biology using 2-photon calcium imaging. And this is what they said: taste qualities in gustatory insula cortex of alert mice is sparse and distributed. And it's the same in human gustatory cortex. And by the way, the data I just showed you has now been replicated by 2 other laboratories. All right, so now we answered that question. We go back to our cognitive question, the one we're really interested in, which is, why does taste-sensitive cortex respond to viewing pictures? And as I just told you, these are the 2 choices. And if it, in fact, it is a specific response, that would be a huge advantage, I think, for the organism, because that would signal not only that there's food out there, but that it's safe versus dangerous. So now we're doing that experiment again. There's Jason Avery, same kind of idea. We're doing the picture food perception task first, always, but now we're blocking it by type. So we got our sweet foods, our salty, our sour, and our other objects for comparison purposes. And now we're doing the taste perception, just as I showed you, sweet, salty, sour, and neutral. So here's our multivariate analysis. Just like I showed you, this is another replication of this finding. Now it's just average across all the pairwise possibilities to show, yeah, we can decode what taste the subjects were experiencing in both left and right dorsal mid-insula. And the same thing happens for viewing the pictures of the food. Again, in other words, we can decode what pictures they're looking at in this same piece of cortex. Okay, so why does taste-sensitive cortex respond to viewing food? Well, here's our answer: to provide taste-specific information critical for survival of the organism. So let me summarize this part. This region, the dorsal mid-insula, contains taste-specific information, that is, distributed patterns of activity for experienced taste and inferred taste when we're looking at these pictures. Now look, everything, of course, is part of a circuit, and this is true. This statement I just said is true for other regions, including, importantly, orbital frontal cortex, which we know is tied into reward and the more hedonistic properties of food. However, that part of the insula was the only region we've seen that was modulated by the homeostatic state of the organism. That is how much glucose was on board. Okay, so now let me turn to this last issue. I just showed you evidence that inferred taste and perceived taste are represented in the same piece of cortex. Well, how do we think about that? What does that mean from a neural perspective? Well, we know what it doesn't mean. It doesn't mean that the neural substrate is the same, right? It can't be. Knowing, imagining, perceiving, inferring, whatever you have, they're different things. So the neural substrate must be different as well. So could be different populations of neurons, and that could be represented by differences at the level of the laminar. And that's where we're going, because now we have a new tool, a really new one, which we're very excited about, and this is layer-specific neuroimaging. And we're going to show you, I'm going to show you evidence of this now by turning to the motor system to look at differences between imagined versus executed actions. And this was motivated by evidence that suggested that primary motor cortex, M1 itself, is involved in both performed and imagined actions. So here's our question. Well, how does performed and imagined actions differ in M1? And two, we get this bonus question: how can imagining that we do all the time influence performance? And you know, there's that vast literature. Anybody who was an athlete out there knows about this, about imagining your performance. You know, I assume most of us imagine our talks before we give them. You practice in imagery. I always go to see like what the podium's going to look like and the stage. So when I go back to my room, I can kind of visualize it. And it's also a big deal in chess. So you remember Beth Harmon in The Queen's Gambit did it on the ceiling, upside down, but it was all through visual imagery. Okay, so what am I doing in the motor system? All of my work has been in the visual system. Why the motor system? Several reasons. The first is it's probably, and I think it is, the thickest cortex in our brain is M1. And in fact, it's like 4 times thicker than V1. So that gives us the possibility of actually seeing differences in the laminar. The second reason is that this has been really fairly well worked out. Input from the cortex goes into the superficial layers of M1, and the deep layers output to the spinal cord so you can act. And then there's a third reason we're doing this, and this has to do with the development of a non-bold imaging method called VASO, vascular space occupancy imaging. This is really the work of Renzo Huber, Laurentis, but he goes by Renzo, in Peter Bandettini's group. And what they've shown, what Renzo has shown over the last few years, that this vaso is really based on cerebral blood volume differences as opposed to bold, and that cerebral volume differences pick up and are dominated by the microvessels shown here in pink that go through all the layers of the cortex, whereas bold is dominated by these large draining veins at the superficial layer. And in fact, based on previous work by Renzo and Peter and his group, they've shown that if you tap your fingers together, you get this characteristic double peak response using vaso, indicating the input from other cortical regions into superficial layers and the output down the cord so you can move. Hope everybody's following. Okay, based on that, we've got a clear prediction, right? When people tap their fingers together, we should see this double peak of superficial and deep layer response. In contrast, when you just imagine tapping your fingers together, we should see activity in, in superficial layers representing input, but no activity in the deep layers relative to our active baseline, which is we just ask people to wiggle their toes. Shouldn't produce any activity in M1. Okay, here's how the study goes. This is somebody's brain. This thing is the hand knob. It's a perfect anatomical marker for where the hand representation is. We drill down into that piece of cortex. Here's, again, high resolution. This is the response in deep layers and superficial layers when people actually tapped. Here I'm showing you the double peak, exactly what we'd expect. And we see when people tap, that's the vaso response. And here I'm showing you that bias towards the superficial layers that I just claimed that one sees with bold. Huge response superficially that just disappears in the deep layers. Okay, we're going to use this now, the tapping, as our localizer. There it is. There's superficial, there's deep. And as I said, what we found is exactly as predicted. Here's the imagining, showing the activity in superficial layers greater than our baseline, where no difference between toe wiggling and imagining in deep layers. And here's the peak, so we could do our stats and show that there is this very robust interaction. Okay, so how do performing and imagining differ in M1? Well, they differ because when you act, you get activity in superficial and deep. When you imagine only superficial, they are not equal. They're not the same, which of course we knew how to be true. We just didn't know how it was represented, even a piece of cortex like this right in M1. All right, so now let's turn to that second question. Well, how does the ability to imagine an action influence your performance of the action? Now look, all I'm plotting here is tapping, nothing else. But what we're looking at now is basically we've divided up the tapping response, the double peak I just showed you, into three components. This is when they tapped, but it was preceded. You get to the punchline. There. This is tapping when it was preceded by tapping. In red is tapping when it was preceded by imagined tapping. And in green is tapping when it was preceded by wiggle your toes. Here's the response in the superficial layers, and here are the peaks, and we get a classic repetition suppression, right? There's your baseline, and both doing the task or imagining the task produces less activity. And you can see the data right there. All right, what's happening in the deep layers? The opposite. We now see repetition enhancement. That is, relative to our baseline, increased activity in both superficial and deep layers. Okay, what's going on? Well, I think what this suggests is that when you are imagining a task, we are actually sharpening that cortical input through repeated imagining, which is reflected in the superficial layers, and at the same time enhancing the motor activity from the deep layers. And that raises, which for us is the million-dollar question, and that is, can you link this to behavior? Why is that a big issue? And it's an issue, by the way, that we've been puzzled about for about 20 years. It's hard to get others to get as interested as we are, but this is why. Look, when you see an object for the first time, like this picture. Hello? Can I keep going? I guess so. When you see an object like this picture of a dog and then see it again, you get this classic repetition suppression phenomena that I just showed you. The puzzle is, how does that make you better? Because this leads to the phenomena of repetition priming. How do you get from reduced firing rates, repetitions, to improvements in behavior? And the other reason we're really always been interested in this is that it's non-hippocampal dependent and it's extremely long lasting. How long? Well, this lag here has been tested up to a year. Something changes when we view objects that appears to produce a permanent change in the processing system. How does that happen? Here's an example. This is our most recent paper trying to understand this phenomena, which, by the way, is preserved from insects on up through us. And we've just tried to understand this by testing all the major models out there, including our synchrony model, Carl Friston's predictive coding model, Rick Henson and Mick Rugg's facilitation model, and the original sharpening model. And we still don't know what's going on, although those of you who are interested can take a look at this paper that just came out. So our hope is maybe these cortical depth findings will give us some insight into how this works, because what we're now seeing was that in the deep layers, there's actually an increase in firing, in which case maybe that is what's correlated with the change in repetition priming. Okay. Let me give you the final summary here. Information about perceived taste and inferred taste are represented by distinct distributed patterns of activity based on that multivariate analysis. We think they're population codes in the same region of insular cortex. That was the story about overlapping neural substrates. Secondly, within that region of cortex, different processes such as performed and imagined motor actions may reflect cortical layer associated differences and their interactions. And finally, cortical depth related investigations should provide a more fine-grained understanding of the relationship between related but disparate processes and to some longstanding questions such as the neural underpinnings of repetition priming and other aspects of cognition. Okay, before I end, let me just take one last minute because, I'm not sure this is the slide I wanted, but on the cognitive level, I've been talking about taste and the insula, but the insula is often also involved in our emotional experiences. And we experience emotion or express it often through metaphors, like parting is such sweet sorrow, right to the bitter end, that's really sweet of you, and so on. And also in a lot of cultures, like here's the traditional Passover plate, and here we eat the bitter herbs to remind us of when we were slaves in Egypt, right? So there's a strong link between taste and emotional experience. Now, I just told you here's a way these things might occur through the interaction in a place like the insula between tasting and information from the cortex. So since we couldn't do that repetition study, we could do something else during COVID and actually explored this using MTurk by having people, over 1,000 of them, look at triplets. This is an implicit task to look at about concepts of color, taste, and emotion and their linkage, and then turn to this cross-linguistic database of 2,474 world languages, 34 language families. I'm not going to go into details about any of this other than to tell you that based on these results, we've been arguing and will argue that taste and emotion concepts are strongly linked, and they're strongly linked not only in English, like parting is such sweet sorrow, but across human language families. All right, let me show you the main people that whose work I just described to you, who you just saw. Here's just a list of the people who were in my section, it's called, not the whole lab, over the last 3 or 4 years, including your own Jason Crutcher, who's in the clinical program here. So as always, it's my pleasure to tell you about their work, and I hope you got some questions.
Thank you very much. Feel free to unmute and clap or do the emoji thing of hi, but thank you very much. That was a fantastic, fabulous talk. Jerry, I'm glad you got to miss all my introduction. I saw you just join as I was finishing. It seemed like you perfectly timed. I was just saying that people are probably timing it to avoid my introduction, which tends to be long. So I try to be short today so we can get a lot of time from Alex. But thank you for recommending Alex. And I know everyone was dying to hear him speak for the past 2 years. So we were so fortunate. And it seems like it was worth the wait because he had fabulous, fabulous recent data that we saw. So we'll get to questions if that's okay. And I'll say the one from the chat box by Tor Wager for now to start with. By the way, I see a comment from BJ Casey, Alex. Also, if you don't see the chat box, she was very much looking forward to your talk, and she has a comment up there. And I'll get to Tor's question. So, hi, Alex. So fascinating. A question for the end. In the Avery 2021 PNAS paper, was there evidence for cross-classification so that taste quality patterns were preserved across states and pictures? And was the organization preserved across participants or idiosyncratic? That is, if you trained a cross-participant classifier, would it work? I had a very similar question, but this is a great question.
So can I share my screen again?
Yes, please.
Because I could do this very quickly. So yeah, that is a great question. And we addressed it. And there, the way I tried to conceptualize it, does experience and inferred taste share similar patterns of activity? So we did it. We did the training with the food pictures and then tested with the taste data and vice versa to answer that question. And and we failed. So here's the data for cross-modal, taste to food or food to taste. Here's our pairwise accuracy, which I showed you before, was high for each of them individually. Cross-modal, nothing.
Wow.
We then did a different analysis, pattern similarity analysis. There again is the taste decoding, there's the food picture decoding, and there's the cross-modal decoding right there. And again, no finding. So why did this happen? Why? Oh yeah, there it is. Why? Okay. So one possibility is that when you look at these pictures, we're getting information about taste, but lots of other properties like flavor, texture, and so on. So my argument is that's just a lousy comparison to do the cross-modal, right? That this is just a lot more than taste. Here it's only taste, this is a lot more. So what's the experiment? Something else we would have done if there was no COVID. Sorry about that. Oh yeah, so instead of doing this with the pictures, let's do the imagining. And by the way, people can imagine, and I'll just try it. You can imagine sweetness, saltiness, and sourness. And now, since it's one property, the question is, will they line up? Can we now go from the actual taste to the imagined taste? And of course, we're going to try this with vaso as well. And I don't have an answer, but I do know, since Jason Avery is doing this, that of the first few subjects we tried the imagining in, you can get activity in this area of dorsal mid insula. So that's my answer. It's a critical issue. People have made claims of this kind of cross-modal decoding. And maybe we just didn't have the power, but I kind of like the idea that it's actually not a fair comparison. So maybe next year I'll have the actual answer.
Great, that's great. Thank you very much. Tor, I hope that was a good, sufficient answer for your question. Yulia. Okay,
Well,
great.
if I can satisfy Tor, then I'm happy.
That's great. Yulia Kovelman from University of Michigan says that it was an outstanding talk. Eileen Yi from UConn, fantastic talk, Alex, comment. And then a question from Eileen. I'm so intrigued by the differences between acting and imagining in superficial versus deep layers of motor cortex, but I'm struggling to apply it to other modalities. Given that perceiving isn't active in the same way acting is, what are your thoughts about analogous differences in, for example, vision?
Great question. And that's what we're doing. So it's not, we did, I hope I made clear that the study that Andrew Persichetti did was really our first attempt. We didn't know if any of this would work. So I went for what there was already evidence for. That is the double peak from actual tapping, motor cortex. So now, Andrew, now that we've gotten back into the scanner, we're in the fusiform. And by the way, unlike motor cortex, we don't know what the laminar organization actually is. We can guess, right, that maybe it follows the same difference between superficial and deep. There's good evidence that it probably doesn't because middle layers are involved. So we're doing the imagining study with objects, famous faces, famous places to start, of course, to find out what actually is going on, and then maybe look at the connectivity, which is the other real main goal here, is what's the connectivity between, not only between a laminar, superficial and deep, but how they interact with the rest of the cortex. I will tell you that way back when we got hold of the 7T, this was the first study I tried, but we knew nothing and we didn't have vaso. And we went to V1 because of Steve Koslin's claims about imagery in V1. Did the right experiment, but we just didn't have the tools, which is why I keep emphasizing that business of the tools. And we just failed miserably. It was just way too noisy to see difference in the laminar. But I understand your question because that's exactly what we're after. And the motor thing was just, oh yeah, okay, we can do this. Now we can get serious about what's happening in areas of the cortex for representing the kinds of processes we're really interested in, like imagined objects versus perceived objects, and try to learn something about what the laminar organization is like in the human brain. So next
Awesome.
year.
Can't wait to hear about it. Bhumika, you're still muted.
I apologize. Thank you, Eileen. This is a question from BJ Casey for something totally different than Tor's question. Do you think environmental context, for example, celebratory meals, family dinners, and so on, play a role in the link between emotion and taste? And an awesome talk as always. I love listening to how you think.
Oh, thank you, BJ. So say that it's so emotion, the way I conceptualized all this, right? It's action, perception, and emotion systems. So my guess, like I'm sure yours, is that our emotional experience is imbued through all of our concepts to one degree or another. And we know so little about this. Oh, there you are. I can see on the screen. And we just don't know. I mean, when I wrote that first grapes paper, one of the reviewers pointed out, you say nothing about emotions. And it's true. There's a vast literature about emotions, but this idea where we started to look at metaphor was really our first attempt to say, look, we know this has got to be a critically important dimension. How are we going to conceptualize it in terms of its role? And, I mean, I agree completely. We look at our animals, for God's sakes, if our emotional reaction is not a huge part of our social cognition, I mean, what is that all about if not our affective response? So we should do a study together, BJ, kind of tackle that.
Great. Any other questions? There's one from Jerry. Jerry, would you like to read the question yourself or say the question?
I'll paraphrase it. First of all, Alex, beautiful talk. So thank you very much for that. I loved it. And I certainly plan on making the recording of this prescribed and obligatory viewing for any of my students. So I wondered whether you've looked at connectivity into the deep layer in this M1 work. So one might imagine, not everyone would imagine this, but some might imagine that there could be some control that is inhibiting the deep layer to prevent action. And I'm thinking, in particular, in something like a go-no-go task, you might actually see this more clearly. There might be some sort of frontal connectivity. I've no idea. And there are other people who might say, no, this is it. That there is no, that is the manifestation of imagination. It's, how that would work, I'm less clear, but the brain is a mystery, as we all know.
So thanks for your comment, Jerry, and your nice words. I can't imagine that they're not interacting. Matter of fact, I was kind of puzzled why you'd ever see such clear responses, superficial and deep and separated. If you remember, I think you were there at Chaos when we first presented this, and everybody was all agitated about why imagining didn't produce motor response, right? And I didn't go into that. We actually measured that, but you would expect to see something down the cord, and we didn't. So it might just be the grossness of the measure. but like I said, I can't imagine that they're not interacting, superficial and deep, because they do. I mean, we know that from neurophysiology, right? So the idea that there's actual active inhibition from the deep layers when we imagine could very well be. I don't, all I can say about that is that Renzo, who's the genius behind all this, who actually is coming back to the NIH, we're very happy about that, has developed procedures for looking at cross-cortical connectivity. If we could figure out how to do within a piece of cortex to answer your question, it certainly would be worthwhile. But I think your intuition is probably correct. There's got to be some kind of active process, but who knows?
Okay, Jerry, are you sure? Okay, great.
I'm super satisfied, always, anything Alex says.
All right, great. So we will
By
move
the way,
on.
Alex, I'm the Jerry Altman, you're right, I'm the Jerry Altman that you did meet at Chaos, I'm not the other one.
What other one? There's only one. Well, I'm very glad that I got the opportunity to do this. I don't like this Zoom business. I don't giving talks this way. I don't know anybody who does, but as I said at the beginning, you know, Jerry and I just kept pushing this back. How could I not? How could it not be by the fall of 2021? I could not be visiting by then. But here we are. So I'm very glad I actually got the opportunity to show you some new data. I appreciate all your questions. It looks like there's stuff in the chat box I could read. And,
Alex, there's one more question before you finish up.
Oh,
Is that
good.
okay if I ask? I feel, if you do have a couple of minutes, I feel bad for not
I
covering
will
this.
sit here for the rest of the afternoon.
All right, thank you. This is from Angela Deach. I know you were kind of wrapping up and I apologize for interrupting, but Angela, I'm not sure you're there if you wanted to read the question yourself or I can read it on your behalf. All right, so let's have it in recording. So the question is from Angela Deach is, thank you so much for this wonderful presentation. Do you, oh, she's in a noisy background, so I will read the question. Do you see any value in the use of visual stimuli as primers to help facilitate neuroplasticity to rehabilitate sensory or motor skills, for example, gustatory or olfactory swallowing deficits associated with long-term COVID?
Oh, thank you for that question. We're about to publish, I hope, a kind of a opinion piece on not just using repetition, but on using covert closed loop neurofeedback, which is another topic I'm really very interested in. And this is an approach to try to, well, to actually reinforce the networks that we think are particularly involved in, for example, social cognition or a particular piece of behavior. We've published on this method and some preliminary data that you can actually not only move the circuits around, which by the way, I think a lot of people have shown now, you can actually move the circuits around in the sense that you can increase the correlation between target regions, which generalizes, by the way, to the rest of the network. So that's been well established. What has not been well established is whether or not you can actually get a change in behavior. We reported in our autistic subjects some evidence that you can. And again, if it wasn't for COVID, we were in the midst of doing a study with people who have poor face memory, attacking the face system through actually reinforcing connections, which we had already demonstrated were weak in our subjects with poor face memory through this covert neurofeedback procedure. So the only thing I can say about this, I'm at the NIMH. I'm really kind of wedded to the mission. I believe in it. And anything we can do through any method that might actually help, we should all be like thinking along these lines. So I appreciate your question, and we're trying.